My primary role is an oncologist or a doctor. At a very early stage I became interested in research, and part of that was the increasing realization that when I saw patients in the clinic and we were discussing treatment for advanced cancer, particularly chemotherapy, what became obvious very quickly was that most patients didn't respond. This is the cell line screening facility.
History is a bit like science. The more you look at it, the more complicated it gets. But I guess there are some key landmark dates that stand out. 1953, when Crick and Watson published The Structure of DNA. 2003, when the first complete human genome is published. But if you go back before 1953, there's an enormous amount of work that tells Crick and Watson that DNA is the thing to be looked at. You might go back as far as the 1860s to the work of Gregor Mendel on inheritance in plants. You might look at the early 1900s with the identification of DNA, the identification of that as the carrier of hereditary information.
A cancer cell line, once upon a time, was a cancer in a human being. And that cancer had some cells taken out of it, and those cells have been enabled to grow in a plastic flask just like the ones in this cabinet, and they will grow indefinitely. You can take, as we have done, over 1,000 of these cancer cell lines, which covers all of the major tissue types that you would see in a clinic, and you can screen those 1,000 cell lines with a large number of cancer drugs. And so in essence, the cell line screen is an attempt to take 1,000 cell lines and to screen them with hundreds of drugs over about 5 years, and knowing the mutations that are present within each cell line to essentially match those mutations to a drug effect. And really, to answer a very simple question, can a knowledge of mutations guide you as to what the right treatment should be in a patient? That is the fundamental goal of this project.
For me, there's one thing which brings the work on genetics and genomics close to what we do in libraries. It relies on the management of huge amounts of data. What you can see behind me is digitization in action, taking records from our collections, turning them into images so that we can put them online. For us, making the archives of genetics available is as important as making the human genome available was in the 1990s and early 2000s. For us, it's not enough just that records are preserved, they have to be accessible so they can be used.
All of what we do within this project is to be made available to the public as a whole, so that any clinician or researcher can go to the website and looking at either drugs or genes can actually ask which gene matches best to which drug with respect to sensitivity. And our expectation and hope is that this will drive a new generation of clinical trials. Clinical trials based very much around sequencing cancers, identifying mutations, and then using that information to define which treatment a patient should get.
In a way, it's quite hard to separate out genetics and genomics from many other areas of trust research, from the other challenges that we face, because of course they impact on our understanding of infectious disease, on chronic disease, on connecting the environment, nutrition and health, and on understanding the brain. But I think there is something particular about genetics, understanding the makeup of our genetic code. That gives it huge potential to impact on these other areas. And for that reason, I think bringing together a range of individuals, a range of perspectives, a range of inputs into thinking through how we might tackle this in future is hugely beneficial for us.
On a weekly basis, what is hammered home to me is how imperfect our knowledge is at present. And it certainly drives home to me how important this type of endeavour could be in treating patients more effectively and moving a little bit closer to this concept of personalised cancer medicine. And I look forward to the day whenever I stop treating cancer patients as if they're all the same and begin dividing patients into groups which are going to be treated more effectively.